Hello everyone, I am Dr. Trupti and this is my YouTube channel Enjoy Biochemistry. If you find the content on this channel useful, please subscribe and like and share the videos. In the video lecture series on molecular biology, today's video is on transcription in prokaryotes. We know that flow of genetic information involves three important processes, that is replication, transcription and translation. So what is transcription? Transcription is the synthesis of RNA from DNA. There is transfer of genetic information from DNA to RNA and it occurs in the nucleus. So the genetic information of DNA is transcribed or copied to the messenger RNA. And during this transcription, the message from DNA is copied in the language of nucleotide, that is 4-letter language. Later this mRNA it reaches cytoplasm where it is translated into functional proteins, and during translation the nucleotide sequence is translated to the language of amino acid sequence, that is 20-letter language. So what is transcription? Transcription is the synthesis of RNA from DNA and it is important for transfer of genetic information from DNA to messenger RNA. Transcription process is similar to replication in many aspects, but it also differs from replication in many ways. So let's first see what are the similarities between replication and transcription. Both processes, replication and transcription, so they are synthesized, like DNA is copied in the replication and messenger RNA is synthesized from DNA in the 5' to 3' direction. Both processes follow Watson-Crick base pairing rules and both the processes have initiation, elongation and termination, and both use multi-component initiation complex. So these are the similarities between replication and transcription. Now let's see the differences between replication and transcription. In the replication, both strands can act as template strand, while in transcription for a single gene, only one strand can act as a template strand. Deoxyribonucleotide triphosphate are required or substrate for replication, having bases adenine, thymine, guanine and cytosine, while ribonucleotides are required for transcription, having bases adenine, uracil, guanine and cytosine. In replication, A pairs with T and G pairs with C, while in transcription, A pairs with U, T pairs with A and G pairs with C. Primer is required for replication, however, it is not required in transcription. The enzyme involved in replication is DNA polymerase, while the enzyme involved in transcription is RNA polymerase. Double-stranded DNA is the product of replication, while single-stranded RNA is the product of transcription. Replication has extensive proofreading mechanism, but transcription doesn't have such mechanism. In the replication, whole genome of DNA is replicated, while transcription involves only small portion of genome. So DNA replication is like printing a copy of all pages of a book. Replication process occurs only at the time of cell division. But transcription is taking place all the time. Only certain areas of DNA are copied, and this is like taking Xerox copy of particular page of the book. What are the various factors involved in the process of transcription? And the first one is template DNA strand. Only single strand of DNA can act as a template strand. Then substrate. Ribonucleotides are required like ATP, UTP, GTP and CTP. So these ribonucleotides are required for the process of transcription. RNA polymerase. RNA polymerase, only one RNA polymerase is required in the prokaryotes, which can transcribe all types of RNA like mRNA, rRNA and tRNA. Then topoisomerase, which will relieve torsional strain developed because of supercoiling, which occurs due to unwinding of DNA. So that will be relieved by topoisomerase. And promoters. Promoters are required for transcription. So the various factors involved for transcription are template strand, ribonucleotides, the enzyme RNA polymerase, topoisomerases and promoters. Let's first understand the terminologies like template strand, coding strand, etc. So this is double-stranded DNA. and this is single-stranded RNA. This is the RNA primary transcript. So if you observe the base sequences in this primary RNA transcript, it is complementary to the base sequence of this lower strand. This is the red one. So this red one, it is called as a template strand. It is the strand from which the RNA is actually transcribed or copied. So if you observe the base sequences, they are complementary to this lower strand, and this strand is called as template strand. Now the upper strand, this is called as coding strand. The strand which corresponds to the sequence of RNA primary transcripts which encodes the product of the gene. So if you observe the base sequences, they are exactly similar to the base sequences of coding strand. Only difference is that in place of T, U is present in the RNA. So the coding strand, the base sequence is similar to the RNA primary transcript, that is RNA, while the sequence of template strand is complementary to the RNA primary transcript. So template strand is nothing but strand from which the RNA is actually transcribed, and coding strand, it is a strand which corresponds to the sequence of RNA primary transcripts. This coding strand is also called as sense strand or non-template strand, and the template strand is also called as non-coding strand or anti-sense strand. So template strand, non-coding strand and anti-sense strand, these are synonyms, while coding strand, sense strand and non-template strand, these are synonyms. Now let's understand the coding and non-coding strands of a gene. Gene is the functional unit of DNA that can be transcribed, and in long DNA molecule, genes are placed in collinear fashion and have either of the two strands as coding strand. So genes may be on any strand of DNA. For example, this is the DNA strand and there are 3 genes. So this is a gene 1 and this is the primary mRNA transcripts, and for this gene 1, this is 5' to 3', the upper strand, it is the template strand, and this lower one 3' to 5', this is the coding strand. For the gene 2 and 3, this 5' to 3' direction, this upper strand, this is the coding strand, and this lower one 3' to 5' direction, this is the template strand. So genes may be on any strand of DNA, and for that particular gene, either of the strand can act as coding strand or template strand. Transcription in prokaryotes involve only one enzyme, that is RNA polymerase, and this RNA polymerase is DNA dependent RNA polymerase, which is multiple subunit protein having molecular weight of 400 kilodalton. It has core enzyme having 5 subunits, 2 identical alpha subunits, one beta prime and omega. And to this core enzyme, when sigma subunit joins, it becomes holoenzyme. And the beta subunit binds to 2 magnesium ions. This RNA polymerase reads template in 3' to 5' direction but synthesizes new RNA in 5' to 3' direction. It does not require primer and it doesn't have efficient proofreading mechanism like DNA polymerase. As I have already mentioned, the RNA polymerase of E.Coli is assembly of 5 important subunits, which is core enzyme, and it involves 2 identical alpha subunits, and these 2 identical alpha subunits joins to beta subunit, which is larger than the alpha subunit, and there is other subunit beta prime and then omega. So now it becomes core enzyme, alpha to beta, beta prime and omega. This is core enzyme, which is represented by the symbol E, and to which sigma factor joins, and now it becomes holoenzyme. So core enzyme plus sigma is holoenzyme. The function of alpha subunit is it is involved in the RNA polymerase assembly. It interacts with DNA and various transcription factors which are required for transcriptional regulation. Beta subunit, it is involved in DNA and RNA binding, in the RNA synthesis, NTP binding. It is the site for rifampicin binding and sigma factor binding. Beta prime subunit, it is involved in DNA binding, RNA synthesis. It is site for catalytic magnesium coordination, and guanosine pyrophosphate binding site 1 and 2, and also sigma factor binding. The omega factor, it is involved in the RNA polymerase folding and guanosine pyrophosphate binding site 1. The sigma factor, it enables core enzyme to recognize and bind the promoter. So the sigma association with core RNA polymerase decreases the affinity of core polymerase for non-promoter DNA while simultaneously increasing the holoenzyme affinity for promoter DNA. So the association of sigma factor to this core enzyme is very important for recognition and binding of this core enzyme to the promoter and to start the transcription process. Promoters are very important in the transcription. They are part of transcription unit. So let's understand about promoters, transcription unit, etc. So this is the DNA strand, and the lower one is the template strand, which is to be transcribed in the RNA. The upper one is the coding strand, and the base sequence of coding strand is similar to the RNA transcript. And this DNA has all the signals required for process of transcription, that is initiation, elongation and termination. The transcription starts at transcription start site, which is called as TSS, and it is denoted by +1 sign. And anything upstream, all the bases upstream to this +1, that is transcription start site, they are denoted by minus sign, like -10, -35, and they are called as 5' flanking sequences. Anything or all bases on the downstream side of this +1, they are represented by plus sign, and they are 3' flanking sequences. Now, how this transcription start site is located by the enzyme RNA polymerase. So with the help of promoter, the RNA polymerase locate this transcription start site. So the promoters, the first promoter region is present at the -10 region, and this has the consensus sequence TA-TA-AT, and this is the 6 base pair sequence. It is called as Tata box or Pibnow box. It is the site of initial DNA melting. So DNA separation occurs here at this -10 region. The second region is -35 region. It is 8 base pair sequence, and it is the initial point of contact of RNA polymerase. So this is the promoter region. This promoter region has two regions, one is -10, one is -35. So RNA polymerase first binds to this -35 region, and then it leads to separation of the two DNA strand at this -10 region. So this is the site of initial DNA separation, and this is the site of binding, and this region it is called as promoter. So what is promoter? Promoter, they are the DNA sequences where RNA polymerase can bind. Now this region, from this transcription start site, it is called as transcribed region, and ultimately there will be formation. The product is primary transcript. It is the RNA product which is synthesized in 5' to 3' direction, and the whole is the transcription unit. It is the region of DNA which includes signals for transcription, that is initiation, elongation and termination. Having learnt about template strand, DNA strand, RNA polymerase enzyme, promoters, transcription unit, now let's understand the process of transcription. It is a cyclic process, and it involves 6 important steps. The first step is binding of RNA polymerase to the template strand and formation of closed complex. Second step is formation of open complex, which is also called as pre-initiation complex. The third is chain initiation. Fourth is promoter clearance. Then there is release of sigma factor. Then chain elongation. And the sixth one is chain termination and RNA polymerase enzyme release. And as it is a cyclic process, it can again restart. So this is the overview of process of transcription in prokaryotes. The first step of transcription is RNA polymerase binding and closed complex formation. So this is the DNA double strand, and the lower one is template strand. +1 is initiation transcription start site. This is Tata box at -10, and at -35 another box, that is promoter region, is there. So at this -35 region, first RNA polymerase enzyme binds with the help of sigma factor. So RNA polymerase binds to the promoter site on the template strand to form closed complex. So there is no separation of DNA strand in this first step. So this is the RNA polymerase enzyme, and with the help of sigma factor, it locates this DNA promoter region and it binds to -35 region, and there is formation of closed complex. The second step is open complex formation and formation of transcription bubble. So open complex is also called as pre-initiation complex, and the RNA polymerase enzyme, it has unwindase activity, and because of this unwindase activity, two strands of DNA, they are separated, and there is open complex formation, and it is called as transcription bubble. So this is the transcription bubble which has 20 base pairs. So this is the second step, that is formation of pre-initiation complex and formation of transcription bubble. The third step is chain initiation, in which RNA polymerase catalyzes the coupling of first base, which is often a purine, though not always, to the second nucleotide base, and there is a formation of phosphodiester bond, and the nascent chain is attached to the polymerization site on the beta subunit of RNA polymerase, and 5' triphosphate end of the first nucleotide becomes 5' end of newly synthesized RNA. So this is how chain initiation occurs. The fourth step is promoter clearance. After RNA chain length reaches 10 to 20 nucleotides, the polymerase undergoes conformational change and then it is able to move away from the promoter. So now the RNA chain is 10 to 20 nucleotides and the RNA polymerase moves away from the promoter region. It is called as promoter clearance, and sigma factor leaves the core enzyme. So this is the fourth step, that is promoter clearance. The fifth step is chain elongation, where RNA polymerase core enzyme progresses along the DNA. It adds successive ribonucleotides as dictated by template strand sequence and according to Watson-Crick base pairing. The pyrophosphate is released following each cycle of polymerization, and this pyrophosphate is rapidly degraded to two molecules of inorganic phosphate by pyrophosphatase, and the reaction becomes irreversible. And the transcription bubble, it keeps on preceding the polymerization, that is, it moves ahead of polymerization with the help of unwindase activity of RNA polymerase. And in the chain elongation, the successive residues are added at the 3' hydroxyl terminus of the nascent RNA molecule until a transcription termination DNA sequence element is encountered. The sixth step is termination, and this termination can be row dependent termination or row factor independent termination. So let's first see row factor dependent termination. Now what is this row factor? It is the hexamer. It has two catalytic activities. First one is ATPase activity and second is RNA DNA hybrid helicase or unwindase activity. Now this RNA transcript, it has row factor utilization site or RUT site, which is rich in cytosine residues, and these sites, this row factor binds, and by binding to this RUT, it translocates in the 5' to 3' direction along this RNA transcripts, and by its helicase activity, it separates RNA from DNA. So this row factor binds to the RUT sequences present on the RNA transcripts. It is translocated in the 5' to 3' direction, and by its helicase activity, it separates RNA from DNA. Second type of termination involves row factor independent signals, and these signals are inverted repeat sequences, and these inverted repeat sequences, they are located 40 base pair downstream to the transcription start site, and they are interrupted and introverted repeat sequences which are rich in GC, that is guanine and cytosine. And these are the inverted repeat sequence, and here at this end there is poly A sequence. This poly A sequence follows inverted repeat sequences, and this is a template strand having poly A sequence. So the RNA transcript synthesized will be having U complementary to this A sequence, and this A to U base pairing will be weak. So because of this poly A sequence and this inverted repeat sequence, the RNA transcript formed will be like this. There will be formation of hairpin loop structure. So as complementary base pairs will be formed in the RNA transcript as per this template strand, there will be formation of hairpin loop structure, and as the poly A sequence, the complementary base is U. So this AU base pairing is weak hybrid which can be easily removed from this template DNA strand. So this is the rho factor independent signals. So in this rho factor independent termination, the RNA transcript formed will be of like hairpin loop structure, and because of this hairpin loop structure, it will not allow RNA polymerase to move further, and the AU base pairing in the DNA RNA hybrid will be easily disrupted, and that's why the transcription will be terminated and there will be release of RNA as well as release of RNA polymerase will occur, and that's how termination occurs. There will be no processing of messenger RNA, that is no transcriptional processing of messenger RNA, but tRNA and rRNA can undergo processing. The regulation of transcription occurs through various operons. It is a transcriptional unit containing regulatory gene, regulatory DNA sequences, that is operator sites, and various structural genes. We will see these operons in the later videos. So now let's summarize the transcription. Transcription is the process of synthesis of RNA from DNA. It is a cyclic process and it involves 6 important steps. In the first step, RNA polymerase holoenzyme, it binds to the template and there is formation of closed complex. So RNAP binds to promoter sites on the template strand to form closed complex. In the second step, there is formation of a open complex because of unwinding of DNA. It is also called as pre-initiation complex, and there is formation of transcription bubble which is 20 base pair long. And then comes chain initiation. In the chain initiation, RNA polymerase catalyzes coupling of first nucleotides, which is often purine, with the second nucleotides with the help of phosphodiester bond. Then various nucleotides are incorporated and there is formation of RNA up to 10 to 20 nucleotides, and after the polymerization of 10 to 20 nucleotides, RNA polymerase physically moves away from the promoter and it is called as promoter clearance. After promoter clearance, the sigma subunit, it releases from the core enzyme. So now only core enzyme of RNA polymerase remains there in the process of transcription, and it is involved in the chain elongation further, and RNA polymerase has unwindase activity, so it causes unwinding of DNA and polymerization also. Topoisomerase relieves the supercoil simultaneously and thus chain elongation occurs till the termination signal is encountered. And the last process is chain termination and RNA polymerase enzyme release, and this termination can be row dependent or row independent. And finally, there is release of RNA polymerase enzyme and primary RNA transcript. And as it is a cyclic process, again the sigma factor can bind to this RNA polymerase and again this process of transcription can start. So in this today's video, we have learned in detail about the transcription of prokaryotes. In the next video, we will see about transcription in eukaryotes. So thank you for watching.